You're not starting again.
No, it started. So I mean,
I wouldn't. I mean, we're gonna. I'm gonna wait for some. Or
it's okay. I mean, I can always cut it.
Lakshmi's on vacation.
so is this everybody?
Yeah.
Yeah, Annie. Yeah, I thought they were wrong.
Yeah, this is okay.
Michelle, you're gonna text Annie and just see. I just left her a few minutes ago. But let's see if she's on her way. I'll just wait one more minute. Okay. I guess we better get started. Welcome, everyone. Today is September 6th, 2017. This is the continuation of the neurophysiology lecture series for Einstein neurology. Today's lecture is going to be on the synapse. You could argue that this is the center of the central nervous system, that everything that we talk about, all of our exams, all of our behavior is really occurring at the level of the synapse. And you could argue that everything else is simply to support the synapse. We're going to build on the concepts of last week's lecture on the plasma membrane and other membrane systems. We'll have an introduction to the synapse. We will talk about the synaptic vesicle and its structure. And then, of course, the central portion, which is the exocytosis and vesicle recycling, along with neurotransmitter synthesis and just general terms and storage and that sort of thing. So if we ask, what is the synapse? The synapse is a communication site between two electrically excitable cells. That's all synapses have that in common. Synapses can be either electrical or chemical. If I just say synapse, you have in mind, I'm sure, the chemical synapse, and that'll be the bulk of our talk. But there are electrical synapses that are very important, and we'll spend a couple minutes on them. You can also have synapses from a receptor cell to the neuron, from a neuron to a neuron, of course, and also from the neuron to the muscle, and that's the neuromuscular junction. There's also evidence that you can probably have glia to glia synapses, especially when you start talking about electrical synapses. To set this in perspective here, we can estimate the number of synapses by assuming approximately 100 billion neurons in the brain, and then assume about 500 synapses per neuron. And that gives you approximately 50 trillion synapses in our brain, which is roughly equal to the number of stars in the known universe. So each of us is carrying around a truly astronomical number of synapses. An electrical synapse is defined by, first of all, that it's a direct electrical connection. There is no chemical intermediary. It's an extremely narrow synaptic cleft. So here's 3.5 nanometers, which is 35 angstroms. If you remember from last week, the plasma membrane was anywhere from maybe 70 angstroms would be a rough approximate size for the thickness of a cell membrane. And so this is half that. Right? So this is very small space. There are no vesicles, there is no fusion, there are no receptors. What you have are gap junctions. And that's what makes the electrical synapses gap junctions. The advantage of an electrical synapse is that it's incredibly fast. So synaptic transmission occurs in 1/10 of a millisecond. And you're going to see that's much faster than in the chemical synapse. The crayfish is a good example. Those giant motor neurons that drive the crayfish tail are electrical synapses, and that flick of that tail is very fast. So that's what you gain when you use an electrical synapse, is speed. You find them in the mammalian nervous system. First of all, cortical interneurons and astrocytes, cortical astrocytes, use gap junctions, electrical synapses, to make syncytia. That is, the glia behave from many points of view as if they are a single organism, even though they're multicellular. And they do that through electrical synapses. So just a couple quick pictures. We've got a lot of pictures in this talk. And here's your presynaptic neuron. You get used to that. Microtubule, cytoplasm, mitochondria are ubiquitous in synapses. And here's your gap junction. This is your electrical synapse right here. And here it is magnified. And you can see that what you really have is you still have bilayers, but the junction between them is extremely small, so that you have these basic ion channels or gap junctions, which meet one another end to end and then form a single pore. And under stimulation of the electrical synapse, then there's flow of ions through the channel. And that's basically it. And here you have the 3D view of the same thing. And that's all that I have to say about electrical synapses. Right? They exist, they're important. You'll see them frequently in the nervous system. They occur in our nervous system, and they are unbelievable speed. What they don't have is any flexibility. So they're very difficult to regulate because they're so simple. So chemical synapses are the opposite. Chemical synapses are amazingly sophisticated, with hundreds of sites at which you can regulate the activity of the synapse. The width of a chemical synapse is 20 nanometers, or about 200 angstroms. So that's roughly 3 times a cell membrane, right? So much wider than the electrical synapse. And of course, you're familiar, a lot of this is going to sound familiar. We're just going to get into more detail. You release the transmitter into the synaptic cleft. It stimulates a postsynaptic cell. And that sounds so simple. But once you start thinking about the complexity of a chemical synapse, you realize how complex this is. You have to synthesize the neurotransmitter. You have to store the neurotransmitter. You have to release the neurotransmitter. It has to get across the synaptic cleft. It has to encounter receptors. The receptors have to start signal. You have to end the signal. And I think that it's interesting to think about the question of how a system so complicated as this could ever have evolved. Right? And part of the answer probably has to do with the transition um, from for organisms like polyps that can exist either as single cell or multicellular. And for them to come together to make a community, a multicellular community, they have to be able to talk to one another and bring each other together. That's the kind of thing. Cyclic AMP is a very primitive neurotransmitter. And as you see it very, very early in phylogeny. All right. Typically, neurotransmitters are small molecules. There are a few hundred Daltons in size. You know them, acetylcholine, glutamate, that sort of thing. We're not going to be talking about individual neurotransmitters today. But you can also have neuropeptides, which are in the 5 to 10,000 Dalton range. They're going to be handled very differently. Right? And initially, back when the synapse was first being discovered, everybody thought the rule was one neuron, one transmitter. Right? And that was believed to be true for a long time, absolutely. And yet, gradually people realized that rule is frequently bent because you can have peptide neurotransmitters in the same cell. And so you don't find a cell which has acetylcholine, let's say, and norepinephrine as a transmitter. You won't find two small neurotransmitters in a single cell, but you will definitely and frequently combine a small neurotransmitter with a peptide. The time course of a chemical synapse, okay, is about 1 millisecond. And if you remember, the electrical synapse was 1/10 of a millisecond. So these are slower, and a millisecond may seem fast, but it's slow compared to the gap junction. Also, just going back here for a second, in addition to the small neurotransmitter, the glutamate or acetylcholine, these synaptic vesicles are packed with ATP, but I mean packed with ATP. And they release that ATP at the time of vesicle activation and membrane fusion. Nobody quite knows what to say right now about the ATP, because it is likely that the ATP is powering a lot of function, so ATPases and that sort of thing. But it is also very likely that, I mean, after all, they're purinergic receptors, right? So it's also likely that the ATP is serving as a modulator or even as a direct neurotransmitter at many, if not all, synapses. So both that they are, the ATP itself is a messenger, and also that it enables enzymes to be active when they otherwise might not be. So here's your basic simple diagram. Here's your cell, here's the postsynaptic neuron, and here are the synaptic vesicles within the synaptic terminal. Here's a magnified view of something we're going to get into in a lot of detail, and that is the synaptic vesicle fused with the plasma membrane of the presynaptic cell. And then the neurotransmitter diffusing across the synaptic cleft and letting ions in through an ion-activated channel, or it could be a G-type protein related channel, which acts through cyclic nucleotide, all right? And then activates the postsynaptic cell. Right. So I was saying to, I guess, Annie or Lorraine, I forget this morning, that, you know, all these things have to be invented. Ventilators and MRIs and CT scans, they all have to be invented. But we also didn't always know about synapses. So when we began with light microscopy, all you saw were these little bulbous structures. You had no idea what they were. And they seemed to be mostly occurring at nerve terminals. And they were called buttons just because of their description in light microscopy, but in French it's boutons. So you had synaptic boutons. Then along came electron microscopy, and you can actually see what was going on, and it's unbelievable. And a large part of today's talk, we're just going to look at synapses. We're just going to look at them, okay? Synapses also, as of 1969, both in Argentina and in the United States, it was found that you could easily isolate synaptic terminals from rat brain just by homogenization and then differential centrifugation. in sucrose gradients, and it was easy. And I did this repeatedly for a long time. And you got this cloud. It was just this hazy blue cloud that was just filled with synaptic terminals, and you could study them all you wanted. And then in the 1980s, the EM became much more sophisticated because you could do histochemistry. So either that you could run chemical reactions, or you could do immune-related staining of the cell and see where various molecules were sitting. In any synapse that you come to, because it is extremely high intensive energy requirement, you're going to have a lot of mitochondria. It is estimated that at least 50% of the energy that's consumed in the brain has to do with synaptic transmission. And if you count the entire process of recycling and everything else, it's likely that at least 80% of all energy consumption in the brain has to do with synaptic transmission. And that's what this is about. That's what's going on in the nervous system. And there are over 1,000 proteins involved in this process. So we're going to look at what it may look like a lot of proteins. I'm going to put up a lot of proteins. And go, oh, that's a lot of protein. That's nothing. Okay, like, all right. So do you mind turning out the lights? Because these are, some of these are my own. I used to do a lot of electron microscopy. So let's just take a look. So here we have a dendrite, and here we have a couple of synapses, synaptic terminals. Actually, 3 of them kind of that. But look, so mitochondria, all right, synaptic vesicles. And remember that when you're dealing with electron microscopy, one way of saying it is that everything you're looking at is an artifact. I mean, everything. And the question is how close it comes to reality. But you have to take EM with a grain of salt. It's an attempt to fix things very quickly, but things are happening very quickly too. And so these, you could look at these and consider these reasonably round is what I would suggest. And sometimes they'll be rounder than other. But then you can also see this density. So here's the synapse, right? This is the junction between the presynaptic terminal and the postsynaptic dendrite. And you can see that this is not simple, all right? First of all, the mitochondria has approached to the dense region. You can see that there is a dense region, that there's densities that extend up into the presynaptic side and densities that extend to the postsynaptic side. And then I hope that you can get a sense that there's striation in here as well. And we're going to look at some of that striation, almost like a railroad track is really what it looks like. The fact that it came apart may or may not be real. It may just be artifactual, or it may be real. We don't know. You have a lot of ill-formed membranes on the postsynaptic side, and those may also be artifactual or may be real. All right. Here appears to be a synaptic vesicle which is budding into the dendrite. Again, maybe you only start to believe that it's real when you see a lot of things occur. Okay, and again, here's another active zone, and that's what this is referred to as the active zone, right? And you can see that there's structure that extends quite a distance away from the active zone. These vesicles appear to be aligned and on some structure that you could imagine, and we will say, is going to bringing them, actually attracting them to the active zone. So all of this is going to be a coordinated whole. Here appears to be some synaptic vesicles which are in the process of recycling, and they are in what we'll call an endosome. Here's a dendritic spine. And look at this dendritic spine has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 synaptic terminals that are in contact with it just in this two-dimensional structure. So it's more than that. All right. And you can see that they don't all appear there are probably several different types. This one's huge, for instance, and they have different densities. Here's a higher mag. Again, you can start to see some of the structure that is going on in the synaptic junction region. You can see the vesicles being brought in. The ubiquitous mitochondria are always there. All right. Again, you can see vesicles that appear to be laying right on the plasma membrane. So they don't look like they're fused yet. They look like they're independent, but they're brought in contact with the active zone. Here's a dendroaxonal synapse. Okay, that's quite definite that this is dendroaxonal. And you can see the synaptic vesicles, and you can see that the postsynaptic density in this case is on the axonal side. Mitochondria. More endosome type arrangements. Another synaptic terminal right close by. Glial cells separating them. Again, you can see the structure within the synaptic cleft. Okay, so don't think of the synaptic cleft as an empty structure. Now, I love this picture and I love this technique. This is called quick freeze deep etch. This is a technique which you'll see several pictures of. And what you do is you take the piece of brain that you're interested in and you put it between two plates which have been cooled with liquid nitrogen. And the tissue, which is wet, sticks and it is immediately frozen to liquid nitrogen temperature. Then you take these two plates of liquid nitrogen and you pull them apart. Just pull, and you fracture the tissue. Okay, the tissue planes you're going to have all sorts of tissue planes that go on after that, and some of them you'll be looking in from the plasma membrane side, and some of them you'll be looking from the extracellular side. And then you take these plates, which is just tissue that's been frozen, and you turn it on end, and you take a gold gun, a gun that's going to shoot gold molecules, gold atoms at the tissue at a low angle to make a shadow, okay? And these are called gold tone quick etch. And that's, see the shadows? So the shadows are gold molecules, gold atoms that have been sprayed onto this to provide relief, okay? So now, and the advantage of this is that this is fast because it's liquid nitrogen. It doesn't use glutaraldehyde, other EM fixative. And so if you see the same artifact, it gives you, because it's two different techniques, you think, well, this is more likely to be real then, not an artifact of glutamate, glutaraldehyde, or a freezing process. So here you can see synaptic vesicles, and you can see the inside of a synaptic vesicle in this case. And you can also get a sense that there are little structures that are holding the synaptic vesicles together, mitochondria. Okay, here's a postsynaptic dendritic terminal, right? And you can see what the inside now of that receptor region looks like, of the active zone on the dendrite side. Okay, we'll come back to this, but I wanted to introduce you to the technique, and you can see the synaptic cleft there. And here you can see so the plasma membrane has separated from the cytoplasm here. That's what's going on here. This is clearly an artifact. Okay. Just a higher and higher magnification, you can now see the bilayer structure of the synaptic vesicle. So they're about 250 angstroms across. And then here, we put this one in. First of all, look at this is a glomerulus-like synapse from the cerebellum. And you can see here, this synaptic, we saw before, a single terminal with 10 boutons. And here we see the opposite. We see a single presynaptic terminal with multiple dendritic contacts, right? And it's filled. This is all filled with vesicles, just packed, and has these dense granules. And the dense granules are the neuropeptide vesicles. Dense core, they're called dense core vesicles.
So
all right?
Axon always full of that. I mean,
So,
how do you differentiate axon dendrite on the selection of concepts?
so if you see the absence of any, the question would be, so first of all, most of the time, if you see vesicles, it's going to be the presynaptic terminal. In the rare case that you have the dendroaxonal, you can tell because the dendrite has the microtubules running through it and the neurofilaments. And you really don't see those in the synaptic terminal. If you think of that initial cartoon, which we'll come back to, the microtubular, and neurofilament structures end before they enter the terminal. And sometimes it can be hard to tell, but this sort of structure here you wouldn't expect to see such a extended structure in a synapse in the terminal. And again, we're just up at very high mag where you can see there's probably 100,000, 120,000 mag, and you're looking at bilayers of the vesicles. I love these. I have just a notebook full of them. And now you can start to see also all of some structure within the postsynaptic density too. And we'll talk about what makes these structures in a little bit. And just lots and lots of pictures. And sometimes they become bulbous on the postsynaptic side. You'll see a couple of these. Again, this is a good one just to look at how these are called docked. These vesicles are said to be docked, D-O-C-K-E-D, docked. They're ready for fusion. This one may even be in the process of fusing right here. What is a synaptic vesicle chemically? It's a lipid bilayer, all right, and it has a lot of protein, all right. The small neurotransmitter vesicles are typically, for the small neurotransmitter, this is, they said 50 nanometers or 500 angstroms. It's a little bit large, but okay. Round, they have a clear center, all right, and they're close to the membrane surface or clustered together. The neuropeptides are much larger. They're 250 nanometers, 2,500 angstroms. They're oval or irregular, and they have a dense core. And you don't usually see them in the active zone. The dense core vesicles, we're going to come back to this, but the release of neuropeptides typically does not occur at the active zone. Okay, the small molecule neurotransmitters are released there. We'll see that. And then you're going to see lots of membrane. They also consist of, the synaptic vesicle has gazillion membrane proteins that are present for fusion and recycling, as well as uptake of the neurotransmitter, synthesis of the neurotransmitter, docking, communicating with one another, and everything else. So this is the same sort of thing we've seen. I don't want to dwell on this at all. Here are these are the clear 25 to 50 nanometer vesicles. Here's your dense core vesicles, much larger. And here's an interesting postsynaptic structure. And again, the mitochondria are everywhere. This is just such an active system. All right, where are they made, that sort of thing. So the vesicles, with all of their intrinsic proteins and enzymes, are synthesized in the cell body. And then they're transported and transported by fast axonal transport, which is, as you can see, 400 millimeters a day. And we'll have a lecture, I guess, next year on axonal transport. And that brings them down to the synapse. Individual enzymes involved in intermediary metabolism are replenished via slow axonal transport, which is really slow. And you can think in terms of 1 to 2 millimeters a day as opposed to 400 millimeters a day. Neurotransmitters are generally synthesized in the synaptic terminal, when it says locally, in the synaptic terminal. And very often they're synthesized within the vesicle itself. So here's your synaptic. This is what I said when I said, you know, it's going to look complicated. This is just a smattering of a few of the proteins. And it's, of course, just an artist's rendering. But you can start to see some of the names that we're going to be looking at in a minute as we start to look at the molecules which are involved in synaptic, all the processes of synaptic neurotransmitter release. Okay. Here's a small molecule neurotransmitter, just in schematic diagram. Notice again that the cytoskeleton ends before the terminal. All right. Neuron, rough ER, Golgi, so there you make your synaptic vesicle. It has proteins embedded. You have inside vesicle monolayer, right? You have the outside monolayer. They have different lipids. They have different proteins, right? They maintain that. There is no flip-flop, so that's it. Once they're made, that's permanent, right? Then you put them in the Golgi apparatus, and if you want to put glucose on it, sialic acids, that sort of thing, you make your glycoproteins, glycolipids. Then you transport your vesicle all the way down, and it hooked on the system via a lot of the proteins and the glycolipids that are attached to it. Then you synthesize the neurotransmitter, fuse with the plasma membrane, release the neurotransmitter, and recycle the protein. And we're going to look in detail. This is not about detail yet. And here are the similar process for your dense core vesicles. This should not be a very active terminal. Now, the presynaptic membrane, okay, of the terminal, the presynaptic membrane, right, is in a hexagonal grid, and we're going to show you a picture of that in a little bit. The synaptic vesicles are brought to the region of active zone, and they are held in place for immediate release. So once the signal, the electrical signal arrives, it isn't that the vesicles have to find their way to the active zone and then release a neurotransmitter. They are already sitting there docked at the plasma membrane, ready to release. Okay? Speed is of the essence. All right. This is just a cartoon. This is just a cartoon showing you what I talked to you before about the quick freeze with fracturing. All right. And this is just showing you that you can have two different surfaces when you fracture, typically at the membrane. It's like pulling this membrane apart. So you're going to be looking down at the lipid bilayers, and one side is going to be from the cytoplasmic side of the bilayer, plasma membrane, and the other one is from the extracellular side. And so the cytosolic side is called the P fracture, and the E is for extracellular. This takes a little bit of getting used to, but here you are. You're looking at the same surface, all right? And what you're doing is we're looking down, or we're looking at the presynaptic terminal, all right? The monolayer that's stuck to the cytosol, right? And what we're seeing here is this array, so right, this parallel array, okay, of these, we don't know what they are. Other experiments tell you that these are calcium channels, actually. So here you have all these calcium channels lined up. Okay? This is in an unstimulated neuron. Then you do the same experiment, but during stimulation, and this is what you end up with. So the simple array is now disturbed. Some of it's still there, but mostly what you see, or you're looking at the inside of synaptic vesicles. These pits are the inside of synaptic vesicles. This is where the vesicles are fusing and releasing their content into the synaptic cleft. Okay? That is, I find that just an amazing thing that you can see that and demonstrate that. Right? And here's the cartoon, which lays out the parallel rays and some vesicles fusing. Here are the calcium channels. Okay? And here's what they're talking about as a hexagonal array. These would be all vesicles which are lined up in the active zone, ready to release your neurotransmitter. And this is the sort of thing that we saw before, and I won't go on. Here's another quick freeze deep etch, and you're looking here into the where the membranes are fusing with the terminals. And I like this here because now you can see this one. I think you can see much better. First of all, here you're looking at the inside of the synaptic vesicle, inside, outside. So when it's convex, you're looking at the outside of it. When it's concave, you're looking at the inside of it. Notice all the lace work that goes between these synaptic vesicles. This is lace work that interconnects them. All right. We've already said that the synaptic cleft is about 200 angstroms wide. So the vesicle is 250 to 500 angstroms in width. Okay, so now I want you to have the firm visualization of all of this. You don't release a vesicle into the synaptic cleft, right? The vesicle is 250 to 500. The cleft is only 200. So the vesicles are not entering the synaptic cleft. They're fusing with the pre-terminal membrane and releasing the neurotransmitter and ATP, okay, and some peptides and sometimes enzymes into the synaptic cleft. The neurotransmitter peaks in this cleft at about 5 millimolar, and it does so within 1/10 of a millisecond. So this is a fast process, okay? So from the arrival of the signal until the release of the neurotransmitter and peaking of the concentration is of the same order of magnitude as the chemical synapse, as the electrical synapse. It's only a tenth of a millisecond. So this is the fast part. The slower part is going to be on the postsynaptic side, the activation of the receptors. The half-life of a neurotransmitter in the synaptic cleft is about 2 milliseconds. Milliseconds, okay, thousandths of a second. And the lateral margins of the synaptic cleft are always closed off by astrocytes. So they're going to, when we look at these two-dimensional pictures, we see what looks like a line. The cleft isn't a line. That's not correct. We're cutting through a surface, all right? So the synaptic zone is a surface. So we're just happen to be looking at a cross-section through that surface. The surface is going to be bound, okay, by glial cells, astrocytes, okay? And the terminal also is a three-dimensional structure, a bouton that's sitting in this zone. And then we take a cross-sectional picture through it, but the whole thing has to be three-dimensional. We already saw that there were pillar-shaped protein complexes which connect the pre- and postsynaptic terminals, and with these vertical rods in the cleft. So we've seen those. Nobody knows why they're there, whether they're structural, whether they hold it together, whether they guide the neurotransmitter and the peptides across. Nobody really knows whether there are anything else in there. The signal ends either because a transporter removes, like the catecholamine, you just remove the neurotransmitter from the synaptic cleft, or in the case of acetylcholine, you use an esterase, okay, and cleave the acetylcholine. Right. And we've seen this picture, but now you can start to have a little bit more knowledge as you start to look at this thing. Maybe you can see again these vertical pillars that are in there. And if I remember right, there was some out here, right? This is an extremely high magnification, probably 200,000 plus. And here again, 200,000 plus. This is the, you don't even see everything else. This is the synaptic cleft right here, between here and here, and you can see the striations between them. This is an unbelievable picture of detail. And you can see that the structure that's involved. I mean, the structure crosses the synaptic cleft, and it goes all the way across here. This is still a structure which is running this terminal area. And here you have all the vesicles which are arriving, fusion, that sort of thing. You almost never see the evidence of a totally fused vesicle. I don't know whether it's because the process is too fast for the animal to catch, but you hardly ever see that. And then the postsynaptic density we just saw extends into the cytoplasm, and that's normal for it to do that. And it creates a web of receptors and machinery that are going to activate the rest of the cell membrane. A very highly organized synaptic cleft right here, active zone, relatively symmetric in this case. And again, you can get a sense of the three-dimensional structure, very, very possibly taking up calcium, removing it, signaling, moving it into its own compartments. And just pictures of just varieties and varieties of synapses. Okay, let's talk about the cycling now. So you have the neurotransmitters are synthesized, okay, so and then transported into the synaptic vesicle. You have to look at the driving force for that. The synaptic vesicles then cluster in front of the active zone and back at the terminal plasma membrane. They become primed for membrane fusion. Calcium enters, it triggers a fusion fork, and then there are lots of different ways to recycle. So let's look at all of this. All right, cartoon. Ending cytoskeleton, here comes the synaptic vesicles. They are now in the bouton. They fill them with a neurotransmitter. All right, so synthesize it in there or take it up into it, whichever you want to do. Bring it to the active zone, all right, and then fusion with the arrival of calcium. And then this would be an example of, so now you have redundant membrane, You have this fused membrane, you can't leave it there, right? So you're going to have to remove it. There are lots, I'll go into different ways that can happen. This is just an example of a class rate coded vesicle that's being recycled. Now you're starting to see some of the proteins that are known to be involved in vesicle recycling. So let's look at a little bit more. You have a reserve pool, right, of vesicles. And then they just live out here away from the active zone in this reserve pool. And the calcium calmodulin kinases and a molecule called synapsin bring them together for what's called mobilization. There's now a group ready. And then they dock. And the docking occurs with GTP binding proteins and snares. So snares were interesting. The term snare doesn't mean very much, but it has to do with a protein which was originally isolated from fungi. Okay, and so the fungi have to be able to both fuse and to cleave themselves. And so they needed proteins to be able to separate and run the membrane portion of the cell replication. And that is, as far as I know, the earliest that these things appear phylogenetically. But then they carry through all the way into the mammalian central nervous system. So typical thing for the brain to do, reuse a protein which has been around since the origin of life. And that's the sort of thing that it does regularly. All right, and then all of these proteins, and I'm not going to get into all of them, are involved in priming the vesicle. And we're going to look at this vesicle fusion and release in detail, and then, like I said, recycling. All right, active transport. Synapsin is the molecule which holds the synaptic vesicles in the reserve pool. All right, synapsin. Calcium calmodulin kinase, okay, triggers their mobilization. All right. So the way that occurs is the kinase phosphorylates the synapsin, and the synapsin releases the synaptic vesicle from the reserve pool. Okay. Then the synaptic vesicles cluster in front of the active zone and dock with the terminal plasma membrane, and this is by GTP binding proteins in those snares. So here you go. So you have a couple of molecules on the synaptic vesicle, like synaptobregan and synaptotagmin. Right? And here you have, notice by the way, we'll look at these even bigger, but notice that they are embedded within the vesicle bilayer. And then you have this syntaxin, all right, which is also embedded in the postsynaptic, in the plasma membrane. And then you have this other molecule called SNAP25, which is interacting with the plasma membrane electrostatically. So again, if you remember back to all the mechanisms that we talked about last week. And then once the vesicle actually docks, you see, this snare complex pulls this together, and this is what it looks like while it's waiting for a calcium signal to arrive. Here's the syntaxin. Here's the synaptobregan. They're intertwined, and now really holding them together and electrostatically bound to the membrane is the SNAP25. All right. Then, so these are all ready. This is what we've just been looking at. I'm not going to say it over again. The only thing that's really interesting is that snare proteins, which again come from fungi, and they're not calcium sensitive. So calcium was not the trigger for those cells to do their things. They were more likely to act via cyclic nucleotides. So calcium is a much later transmitter, if you will, or activator. So the proteins which come from the origin of life level, okay, are not really calcium sensitive. And so you have to put on other molecules which make them calcium sensitive. And here you are now, sort of at the molecular level, a alpha helix that goes through the plasma membrane, an alpha helix that goes through the synaptic vesicle, and the SNAP25, which is doing an electrostatic interaction with the head group and is very active in the vesicle fusion process. Now, nobody really understands the fusion process when you really get down to it at the individual lipid molecule layer. But here's a good cartoon of the sort of thing that must occur. So here, this is the complex that we were just looking at. You have a couple of these now, probably quite a few more that are going to be in a circle. Again, think three-dimensional. We're going to make a 4. So watch what happens. All right? These things basically start to clear away, and almost they almost look like they act like scissors. So they embed themselves in. They take some of the lipid molecules, which are making these two separate bilayers, and they remove them. They just simply pull them onto themselves, onto the molecule, onto the protein. And when they do that, they open a pore between the vesicle and the plasma membrane, like this. So this is as good a cartoon as you're going to see on how it works. And it's all triggered by calcium. So the calcium uses a protein called complexin, which clears the proteins in the area where the vesicles are going to fuse. So they have nothing but lipid now. So the first step in fusion is you take all the protein away from the bilayer. And what you have are two lipid bilayers approximating one another. All right? And then under the influence of the calcium, these things are triggered, and you can just see the sort of thing that's happening. Right? And creating this area. And then it's going to have to let it re-anneal and let this re-anneal. Nobody knows how these things really happen. And then what's going to happen to this piece of membrane, which it cut out, and because it had a asymmetry, and it can't just be reinserting it into the membrane, so that doesn't work very well. So nobody knows what happens to this area, those lipids.
It's supposed to, if you really use it out and snare, should go like this.
Yeah, and yeah, I mean, but yeah, do you make the plasma membrane bigger? I think not. Would you want to destroy the molecule? I think not. I think you want to use it. You're a long way from the cell body where it's going to be synthesized, so that wouldn't be the most efficient thing. And then you have to also think in terms of speed. The speed at which these synaptic vesicles are fusing and generating excess membrane. And this is what we've seen, and now you can see again the vesicles from the inside. And this is what we've seen. And another cartoon just to show you the same thing. This always strikes me as very funny. This looks like someone who took the emergency door off the plane. You know, they had to they pick up the door, and it's just these two people standing there having opened this up. Here they are bending over, and then they're lifting it up, a shield or something like that. And anyway, the idea is that it fuses and releases the neurotransmitters immediately. This is probably a better example of what it actually looks like. Although some of the EMs suggest that you really do do it that way. Finally, we get to recycling, and there are lots of different ways to do it. There are basically 3. The one is where the vesicle just simply, it's called kiss and run. It opens up a very small pore, it re-anneals the pore, and it floats away. And that's that. And it's ready to go again. So that's called, this one's called kiss and run. So the question is, do you recycle it? Does it stay in the active pool? If it stays in the active pool, they call it kiss and stay. If it's recycled away from the active zone, then it's kiss and run. Or you can use Claflin to take coated pits and remake synaptic vesicles. So this is just a cartoon, kiss and stay, kiss and run. And, really, it's an endosomal recycling using Claflin. And here's Claflin. And here's this has this amazing soccer ball-like structure, this triskelion. And that creates this cage which surrounds the synaptic vesicle and returns it to the endosome for recycling. Here's the EM version of that, and it looks like a kid's cereal, honeycomb, so that sort of thing. But these things are real. That's what continuously blows my mind, is that these cartoons, which look so strange, the EM shows you that they're real. These are really there. The way that the neurotransmitter gets pulled into the vesicle is via a proton gradient. So the main ion transporter of the synaptic vesicle is a proton ATPase. And that's what I would say. There is also a chloride gradient which may be used. So it's really acidification, and that's what has to occur. And this occurs very, very quickly. The proton ATPase sits on the vesicle membrane. As soon as it releases itself from the plasma membrane, it's almost immediately reacidified immediately. And this is just talking about, again, here, I want to focus on all the ATP that is stored within neurotransmitters and complexes. I think that's enough for the cartoons. There's a molecular diagram showing a complex of ATP with acetylcholine and linked via a sialic acid, interestingly. Just to go over one more time to this release process, some of the things that happen is we're going to talk about calcium signaling. Calcium influx goes from you're in the millimolar concentration extracellularly, right? You got 1 to 2 millimole at least standard, right? And then here you have on the inside you have 4 orders of magnitude lower. So any calcium that enters the synaptic terminal is going to be noticed. This is a very low level of calcium. And then I think we've been over all of this. The only thing here from this slide that is new is that in order to release peptides, in order to activate the dense core vesicles, which have peptides in them, you have to have a sustained level of elevated calcium. So these quick bursts of calcium, which are typical of neurotransmission with small molecules, won't activate the peptide. In order to activate the peptide, you basically have to be doing something like long-term potentiation. You have to raise the calcium level and keep it up. Okay? That's when the dense core vesicles start to move to the membrane surface. And that's what this cartoon is showing. And they also show that they tend to occur away from the active zone. The vesicle fusion of the dense core vesicles is usually not here. It's out there into the extracellular space. And notice again, I talk about LTP, so here's a high frequency stimulation. The standard low frequency stimulation will not activate this system. So learning may have to do with this sort of. And then termination, I think you have a sense of, you've been through this, esterases. Okay. And that's a lot of detail about synaptic transmission, but that is, I'll say again, that's what makes us tick. That's what this is all about. And everything else is to support this and make this happen. Okay? All right. Good.
Yeah, that was just awakening. So